Yeah, welcome, welcome. It's a great pleasure to be here. Well, I think 2 degrees, it's 2 degrees too hot, so you might want to work on this. And we are here at the Biennial, and it's a very important point in time for Instruct because we face our next 5-year cycle. So every 5 years we have to go back to our funders, to our ministries, and make the point why integrated structural biology is important and why we ask for their support. And so my contribution will actually tell you a little bit about what is Instruct, the entire Instruct community, and contributions by the community, and then also at the end some of my signs. Instruct funds access for researchers. It is as easy as this, in a pan-European manner, in a manner that at the foundation of Instruct, it was clear that we have to move from isolated structural studies to integrated structural biology studies using all the modalities in structural biology. And this currently happens in 29 facilities that provide 102 services in 9 technology types, and we integrate as we speak new modalities over this platform. So it's a constant adaptation of new technologies that goes on in the centers, in all the countries, in Instruct. So if you look at this for a year, that's not, 2024, then there were 271 research proposals received by Instruct, evaluated, peer-reviewed, leading to 179 access visits, 338 publications, and this is all steered or managed through the ARIA Research Infrastructure platform with 21,000 registered users. So this is, you could say, this is part of this community, but the community is even larger, but this is one measure for community. Now, if you look at impact, one way of how we look at impact is, of course, citations. And I did this analysis yesterday night, and it might not be entirely correct. The numbers may change by plus minus 5% or so, but last night, Web of Science core collection using Instruct-ERIC in all fields gave to me 1,500 publications, and since Instruct-ERIC, this brand was started, and they contribute to 10 Sustainable Development Goals, with the vast majority of the publications contributing to SDG 3, which is good health and well-being. And if you look at this, Regina Günster made this slide showing that, of course, we know that, that our medical colleagues characterize contributions towards disease indications or mechanisms. And so Instruct contributes basically to everything that's in medicine. And we argue, as structural biologists, is if you want to understand the mechanism, the reason for disease, then you have to understand the structures, because then you can target these diseases. Now, on our web page, we try to monitor excellence as documented by publications arising from Instruct. And so this is the category scientific highlights defined under science. And I now go through very quickly some of the highlights that we have featured and invite you to reach out to us. I mean, we reach out to you, but you can also reach out to us. If there is a great paper from your lab, we are very happy to feature your work. And so one of the things which were the highlights of February and April this year is 2 contributions in computational structural biology with metal binding site identification and the version 3 of HADOC, and that comes from Instruct Netherlands and Instruct Italy. We have a highlight in March of this year where new technology in cryo-ET is being developed through a research and development grant, comes from Pletsko in Martinsried. And then Instruct France, November 2025, has a high excellence in understanding enzyme catalysis that was funded through Access to Instruct France. We have excellence in biomaterials funded through Access to EMBL by colleagues from Greece, understanding the mechanism of beta-glucosidase. We have, again, excellence in combating antibiotic resistance from Markus Weingart, teaming up with Instruct Italy to determine the structure of these antibiotic active compounds, and contribution to our Belgian host. This is a really nice, a somewhat earlier contribution, is the use of nanobodies in trying to understand GPCR signaling. There's an X-ray structure, there's a cryo-EM structure, and that was a collaboration between University of Oxford, Diamond, and Jan Steyer's lab. Now, with these contributions, where we try to highlight the contributions in the field of integrated structural biology, we also think we have to reflect on the future of integrated structural biology. It is our duty as scientific community to actually put forward what we believe is important. In 2024, we published such a community paper. You know these papers, they have lots of authors, and the advantage of this is that you try to get a consensus view over different technologies, different fields. And another advantage is these things tend to be well cited, because you duplicate the communities that cite these articles. And in this, at sort of the end, we conduct a 5 forces analysis. I mean, this is not, we would all underwrite this, so it is not entirely deep, but I'd like to point out that the frontier 1 on the top is sample preparation. Whatever we do needs a sample, because we are experimental scientists, most of us. So the sample preparation and the access to sample preparation for people that are not so experienced making very difficult samples is a key asset of Instruct. Then, of course, we try to move into in situ cellular structures. We try to understand structural dynamics, intrinsically disordered proteins. We try to actually translate such insight into translation biology, and all of this faces nowadays AI. And so our next spiel on such a community paper will be on AI in structural biology, which is accepted for publication in NSMB that will come out soon. Now, in this article, I'd like to emphasize a point where I believe structural biology also goes, and where access to the research infrastructures will help researchers to do different structural biology. So we know that it was very difficult to solve the first GPCR structure, but now this is a gallery of 403 structures of GPCRs. And it is the sum of those structures, or the ensemble of those structures, that actually faithfully map out or make us understand how GPCRs work. And again, Jan Steyert actually, with his antibodies, tries to stabilize antagonist, agonist, partial antagonistic states. And if you look at contributions by Stefan Grzelczyk, then he sees how you can actually map out the response of such GPCRs by NMR as you add small molecules. And this is a very specific response for each of the small molecules. So it is the sum of the structures and not the individual structures that is part of the future of integrated structural biology, and for this you need to have the resources to conduct many structural studies. And also this is important for the field of drug discovery. And Frank von Delft gave this presentation 2 weeks ago in London, and he allowed me to share his slides because he is really a driver in the X-ray part of fragment-based drug discovery. And so we all know the concept, or many of us know the concept, and Frank summarizes this. HDS, a high throughput screening, is hope for luck, and fragment-based screening is make your luck. Of course, you start with low affinity compounds, and yet you have to believe that chemistry will allow you to optimally link these fragments, but it is a structure-based method of optimizing compound classes. So, and we know that growing, merging, and linking as strategies, but this is the real challenge with fragment progression. Learning by doing. And he also announced the fact that Diamond has secured new funding to increase this KO4 flagship beamline in 2029-30, which is part of our next 5-year cycle. And he projects that there will be a factor of 10 more screening capabilities, and that goes into, it is the sum of all the structures that give you insight on how to develop the drugs, and it's not an isolated structure. So you have to have the capacity to run those in big numbers. And as an NMR spectroscopist, I think I can say that being able to screen a 1,000 library overnight and get the 30 crystal structures of the fragments binding to a biomolecular target is truly amazing, and we hadn't foreseen this 10 years ago, or even probably 6 years ago. This kind of work under Frank actually accumulated in this COVID moonshot, where they developed a main protease inhibitor that became preclinical in 2025. Now, as Instruct, we also contribute to this. As Instruct community, trying to gather funds from the Commission in tech development projects, and the project that we are leading is called Fragment Screen, that joins many industry partners, Bruker, Thermo Fisher, IBM, but also our friends in EU OpenScreen, because we need help from medicinal chemistry. And we try to develop the technology and the instrumentation and the computational workflows to actually do this fragment progression. Now, at the last 10 minutes of my talk, I'd like to welcome you or invite you to think about whether not only proteins can be targeted, but there's a second class of biomolecules. Sometimes you have to tell this to friends. They are called RNA. They have something like 10% of PDB space or PDB deposition space. And so why you want to target RNA? And Kevin Weeks, who is the person that invented SHAPE, that is a method for 3D structure determination, has written 2 opinion pieces in Nature Reviews Drug Discovery and Current Opinion in Structural Biology, which are to the topic. So if you look at, we all know that it is a minute fraction of the transcriptome that is being translated, something like 1%. And out of this 1.5% that is only translated into proteins, you probably have only a fraction that can be targeted by existing drugs. And then you have a large portion of the proteome that is very difficult to target. However, if you take the other 99% of the transcriptome, which is the non-coding RNAs, then he makes a prediction that 70% of those transcribed but not translated RNAs, the non-coding RNAs, could potentially be targeted by a small molecule. Now, maybe this is an exaggeration, but if it even was just 5 or 10%, it would be a big thing. Now, and then he, in 2023, he comments on innovations by targeting RNA by fragment-based ligand discovery. And you see here the starting fragments and their fusion into linked sort of hits. And you go from 2 times above 100 micromolar affinity to 6.5 micromolar, 62 and 550 to 44, and so forth. And this is work conducted in my lab, starting with the DSI poised library developed by Frank and others in the iNext Discovery project, where we started with 90 and 160 micromolar affinity and went now to something like 1 micromolar affinity. And if you look at the technologies that are being essential for this initial fragment screening to RNA, then it is mass spectrometry and NMR that were used to actually get those. Now, in order to then translate this initial affinity ligand small molecule binding to RNA, you have to solve the structures of RNAs. And we know the pictures of the proteome of SARS-CoV-2, and this is the picture of the transcriptome of SARS-CoV-2. I dare say that all of the structures have been solved in my lab, and we use NMR, and we use SACs in Hamburg and in Grenoble to actually get shapes and use SACs to cross-validate our more highly resolved NMR structures. And you see why it is difficult to target RNA and to do structures is that RNAs also have these IDRs, these intrinsically disordered regions that are also very difficult to understand for proteins. So we focused on trying to develop small molecules that target the frame shift, the programmed -1 frame shift element in SARS-CoV-2, which is a part of the coding part of the RNA, which actually triggers the amount of polyprotein 1A over polyprotein 1AB. And the longer polyprotein containing 16 non-structured proteins contains the polymerase, while the shorter one contains the main protease, the 2 proteases and the main protease. And it was early on reported that reduction of frame shift efficiency has a negative impact on viral replication. So the idea is to stabilize or destabilize this pseudo-knot element that forms a roadblock towards translation and thereby modulate the expression of the 2 alternative polyproteins. Now, the work was conducted mainly by Jenny Adam and Betül Seiler. When you start out, you have to say, what is our current 3D structure model? And these were the 3 structures of the pseudoknot, 2 by Caillot and 1 by crystallography, all of which are of course exact, but the ones that is most relevant, I mean they're different, markedly different. The one that's most relevant for our studies, as evidenced by SACs and shape, is this Nenad-Bahn structure that then we used as a model for the target. And I'd like to point out that part of the difficulty in RNA structure is that depending on how much free mRNA you have that is not yet engaged in interaction with ribosome, you have shifts in secondary structure. So our target here is a 69 nucleotide long RNA, but if you look at an 84 nucleotide long RNA, you have a shift in structure such that structure in RNA is a function of the length of available transcript that is available in the cell. So it is not one structure. It is a sort of a dynamic target structure. So this was the lead that we had from our screening, and then we derivatized very simplistically the north and south parts of this DO1 lead structure in chemistry that a medicinal chemist can immediately grasp how you do this. So this is kind of straightforward chemistry. And we actually then have a series of compounds where this HSJA1063 is active in mice against SARS-CoV-2, which is nice. Now let's go into looking at the structural aspects of that. And so we have this compound that has this 6-membered ring, the piperidine. And piperidine, many people don't care, can be protonated at a pKa of 7.3. And we show in a minute that it is the protonated form of this compound that is actually binding to our RNA. And the protonation actually triggers the oral availability of the compound as it traverses lipophilic environments. So knowing the pKa of that compound, which is a common part of a drug, is extremely important. And so you see here that this ring undergoes ring inversion, but also here's the protonation. If you protonate 7.6, then the nitrogen inversion is hindered, which actually restricts the entropy contribution to free ligand substantially. Now, our bound state is in slow exchange with the free state, and by NMR we can determine the off and on rates by exchange experiments, and we can determine the structure of the bound form. And here I summarize that structure. It is basically targeting a bulged out A in the protonated form. And we have cation-pi interaction, pi-pi stacking, and van der Waals interactions that stabilize this conformation. And with this, I'd like to slowly come to my end and say, if you want to conduct great science and want to be supported in your scientific career by internships, if you want to learn more about the latest trends in structural biology, if you have a great idea where you want to get seed funding to develop this idea, or when you want to use our facilities, this is the portfolio of services that Instruct-ERIC over European countries in an integrated manner gives to you. And with this, I leave this on so you can scan it. And with this, I'd like to acknowledge my group, and I'd like to thank you for your attention.